Hello everyone. Today I would like to present my topic about the effect of natural product on enzyme activity as an anti-diabetic potential treatment. In this topic, we will discuss about overview on starch breakdown, diabetes mellitus type 2 and current treatment, and then overview on enzyme and alpha-glukosidase, then overview on natural product and effect of natural product on alpha-glukosidase. Overview on starch breakdown. As starch enters mouth, alpha amylase initiates the breakdown of starch into smaller pieces. It enters stomach where it mixed with gastric liquid that has extreme low pH, which causes denaturation. As it enters small intestine, alpha amylase from pancreatic cell and alpha-glukosidase complete the starch breakdown into glucose. As the glucose enters the bloodstream, it will increase the glucose level in the blood. This triggers the insulin production in the beta cell of the pancreas. Then the insulin is directly flowed through the bloodstream to the organ cells in the body and will eventually decrease the sugar blood levels back to normal. Diabetes mellitus type 2. Diabetes is a disorder that causes disturbance on glucose metabolism. The most common cases is diabetes mellitus type 2. Diabetes mellitus type 2 is a metabolic disorder which caused by the insufficient insulin production or insulin resistance. In normal case, insulin is produced by healthy beta pancreatic cells in a normal concentration. Then the insulin will enter the bloodstream to all parts of the body. As the insulin reaches the cells, it will bind to the insulin receptors. This will cause the nutrient channels open and will allow the glucose to enter the cells. Factors that can contribute to diabetes mellitus type 2 are intensity of physical activity, genetic mutation, diet, and age. As insulin binds to the receptors, it will cause a series of reactions. This reaction will eventually open the nutrient channel. As it opens, the glucose will enter the cell. In diabetes mellitus type 2, if there is an insufficient production of insulin in the beta pancreatic cell due to the cell damage, only small amount of insulin will be synthesized and flowed through the bloodstream. The lack of insulin can cause nutrient channel to stay closed and prevent the glucose to enter cell. If there is an insulin resistance, the cells will ignore the presence of insulin. Because of this, glucose cannot enter the cells for glycolysis process. The current treatment for diabetes mellitus type 2 are present in this PowerPoint. The current drug acarbose inhibit the alpha-glukosidase on the active site of the enzyme. This will slow down the absorption of the monosaccharide into the bloodstream. This will also help to regulate the blood sugar. Enzyme is a biological substance that catalyzes chemical reaction in living organism by lowering the activation energy and without being altered. It is generally made up of amino acids that are chemically bonded by peptide bond and folded, forming three-dimensional structure, except for ribosome. It is specific towards the substrates, meaning enzyme can only react with specific substrates. Enzyme's nature is to reduce the activation energy and increase the reaction velocity, shown in this diagram. There are three types of inhibitor. First is competitive inhibitor, is when inhibitor binds to the enzyme active site. And then non-competitive inhibitor is when inhibitor binds to other side of the enzyme and causing the active site to deform. The third is Uncompetitive inhibitor. When inhibitor binds to enzyme substrate complex and disturb the enzyme activity. Alpha-glukosidase. There are a lot of enzyme that has been discovered in nature and has its own function. Alpha-glukosidase is one of many enzymes that catalyzes the breakdown of oligosaccharide into monosaccharide for absorption. It is located on microvilli on small intestine. It cleaves 1/4 glycosidic bond. The activity of this enzyme on diabetes mellitus type 2 patient can worsen the glucose concentration in the blood. One way to overcome this is to inhibit the enzyme activity. Natural product is defined as any compound that is synthesized in microorganism, plant or animals by specific enzyme. There are two forms of natural product. First is primary metabolite. The compound which plays a vital role act as building blocks or for organisms for growth. Secondary metabolite, the compound which is not essentially used for survival or life of the organism. Both of the metabolite can be used as a drug precursor and other commercial use. Here are the groups of natural product, which are flavonoid, polyketide, phenylpropanoid, terpenoid, and alkaloid. So the question is, can natural product inhibit alpha-glukosidase? There are many research that confirms the potential use of natural product, and many of them have the ability to inhibit the alpha-glukosidase activity. Here are the examples of natural product that shows the inhibition. First is gallic acid and quercetin. Both of the compounds are synthesized in nature and has several bioactivity, including enzyme inhibitor. Gallic acid is synthesized in nature via shikimic pathway, which utilizing the erythrose and 6 enzymes. Quercetin is a flavonoid compound that is usually found in plant tissues and sometimes form complex with other compounds. The computational or docking analysis of gallic acid against alpha-glukosidase has been done by a group of scientists. Here, diagram on the right side, we have amino acids surrounds the gallic acids, which represents the part of the alpha-glukosidase. In this case, interaction occur between serine and tyrosine and gallic acid. In this case, hydrogen bond involved in the interaction. In vitro study shows that pure acarbose has stronger inhibition effect than pure gallic acid. In this research, acarbose is used as a comparison to gallic acid. Here we have diagram which represents the sample 1 until sample 5. Sample 1 is pure acarbose, sample 2 is pure gallic acid, sample 3 is equal combination between acarbose and gallic acid, sample 4 is 75% of acarbose and 25% of gallic acid. Sample 5 represents 25% of acarbose and 75% of gallic acid. The equal combination between acarbose and gallic acids, sample 3 shows stronger inhibition effect than other combination, but slightly weaker than pure acarbose. Quercetin also gives an inhibition effect against alpha-glukosidase activity by computational or docking analysis. In this case, the interaction to aspartate residue and glutamate residue occurs, and it forms hydrogen bonds. In vitro study shows the inhibition value for quercetin. IC50, or it stands for inhibitory concentration, shows the concentration of a substance needed to inhibit the specific cells or enzymes activity by 50%. Low IC50 value will show stronger inhibition effect. In this case, quercetin shows stronger inhibition effect than arquebus. In other words, natural product has the potential to inhibit the alpha-glukosidase activity. This will prevent the conversion of oligosaccharide to monosaccharide. In conclusion, diabetes mellitus type 2 is still a threat towards human health, and it is caused by insufficient insulin production or insulin resistance. Both insufficient insulin production and insulin resistance can increase the blood sugar concentration. Natural product gives the inhibition effect against alpha-glukosidase activity, which may slow down the glucose uptake into bloodstream. This helps the blood sugar regulation. These are the references used for this presentation. I think that's all from my presentation. I hope you enjoy my presentation. Thank you.